Good evening, ladies and gentlemen. For those who don't know me, I'm Peter Joyce. I'm Dean at the University of Otago, Christchurch. Every autumn we run a series of public health lectures on a whole variety of topics. Tonight's the second in a series of 8, and tonight it's made my great pleasure to introduce Professor Patrick Sullivan. So Patrick did his medical degree in the United States, began his training as a psychiatry in Pittsburgh, but actually completed it in Christchurch. And he was on the staff of the University of Otago, Christchurch here for a number of years, before he received an offer from America that we could not match. And so Patrick returned to North America. Currently, he has 2 jobs, one at the University of North Carolina, where he's professor of both psychiatry and genetics, but he also spends half of the year in Sweden at the Karolinska Institute, where he's also got research groups. And over the years since leaving Christchurch, he's been an incredibly prolific researcher in a whole variety of fields around genetics and psychiatry. And so it's my delight to invite Patrick to come and talk about schizophrenia and genetics, and we're very happy. Thank you, Patrick.
Thank you very much. It's an absolute delight to be back here. I think it's been about 22 years since I last stood here giving a talk. I hope this one will be better than the last one, because the last one didn't go so well. This is an image from, I think, 1992, January, some West Coast beach in the summer. That's me with my son Brendan. Obviously, there's a few more gray hairs now than before. The one thing that Peter did mention is that my start in genetics actually happened here, and I was I was a research registrar for Peter in the early 90s, and then we started in this department, and support from the HRC actually made that happen. I owe Peter a huge debt. Really, a lot of what I am as a researcher began under his tutelage. A lot of the bad habits I had, he very quickly got rid of, and certainly helped me enormously along the way. I also like to thank Martin Kennedy, who was really the first person that I ever started talking about molecular genetics with. And Martin proved to be incredibly patient with answering my very, very many stupid questions over time. Okay, today, schizophrenia genetics. 4 parts to this big picture: what we know, what's coming, what does this mean. The first 2 are part of the update in the title of my talk. The last 2 are about the agenda for the next 5 years. Let's start with the big picture. Schizophrenia. Lifetime prevalence around 1%. So about 1% of people in a population will get schizophrenia in the course of their lifetime. It's characterized very briefly by persistent delusions and hallucinations of unknown cause. When I say persistent, it's not just having a bad day or a bad week. It really is at least 6 months of active illness, along with delusions and hallucinations which are very, very far out of the norm. In terms of its impact, schizophrenia has a massive impact on public health. In terms of morbidity, which is sort of the non-death-related ways in which an illness can mess somebody up, it always ranks among the top 10 in the world. In terms of mortality, actual death, people with schizophrenia tend to have 10 to 15, life expectancies 10 to 15 years less than average. So especially in an industrialized country, that's a massive difference. And then finally, the personal, familial, and societal costs are enormous. If you want to put a number on it, it comes out to something like $1.4 million per person per lifetime. So it's a huge, huge societal problem. Now, scientists have been looking at schizophrenia for 100 plus years. And every generation of scientists has applied the best ideas and the best technologies that they've been able to do. And things that worked really, really well in many other areas of medicine have not really worked well for schizophrenia. So the typical methods for epidemiology, for infectious disease, for neuropathology, et cetera, every generation of scientists tried to crack this nut, and it's been very, very difficult to do. I think at the very start, though, I would point out that one of the things about schizophrenia that's crucial to keep in mind is that it probably is due to a fairly subtle process. Many people with schizophrenia actually develop normally for a couple of decades, and then, even before we had medicines for the disorder, people would tend to get quite ill, and then they tend to get better after a period of time. They have this sort of waxing and waning course. And even when people were at their most ill, the basic neurological function was intact, basic cognition was intact, sensorium was intact, etc. It wasn't as if once the illness began, there was this rapid, inexorable decline to coma, seizures, and death. And so I would argue that from the beginning, just from a clinical perspective, a subtle process is likely. In terms of genetics, this has actually emerged as a major clue from a couple of generations of scientific study. But the key thing is that the genetic factors are not deterministic, they're probabilistic. So we're not talking about something like when people hear about genetics, they think, aha, if I have that, then I'm going to get some outcome. It's not that. People who are at high risk for schizophrenia, for whatever reason, they have a subtle increase in risk rather than something that's marked. The risk might go from 1% to something higher, but never from essentially 0% to essentially 100%. So, family history. People with family history of schizophrenia, or a person with schizophrenia, if you look at all their immediate relatives, those relatives are 10 times more likely to have schizophrenia. But that's a relative risk. The absolute risk, though, is far, far less than that. So if the lifetime risk is 1%, the family cluster around a person with schizophrenia, maybe their risk is something like on the order of 8, 9, 10%. So again, that implies that most people with a family history of schizophrenia simply don't get it themselves. And then there have been twin studies that have looked at this for a long time, and even when one identical twin has schizophrenia, the risk to the co-twin is only about 50%. So again, clearly there's more than just a genetic factor happening here. And we've looked for a long time, and there really have been no single gene causes. And again, I would argue that this is consistent with a rather subtle biological process. So given that genetics is, however, a reasonable clue into the etiology of schizophrenia, and given that most other things haven't worked very well, okay, why would we do genetics? What's the point? What are the goals? I would argue that there are essentially two major reasons to do it. I like this sort of primary, secondary, and tertiary prevention. Primary prevention is when you try to prevent a case from happening in the first place. Can we do something to stop schizophrenia from developing? Secondary prevention is when a case is formed, when somebody has an onset, to minimize time from developing the disease to actual treatment. That's not really a genetic problem. That really is more having to do with treatment services, detection, etc. But tertiary prevention is certainly something that genetics can help with. Can we use genetic knowledge to help us figure out better therapies for particular people? And for the primary and tertiary ideas, a really good, a really precise knowledge of the biology of schizophrenia is absolutely essential. And that's the thing that genetics can deliver. Now, going back about 9 years right now, the field of psychogenetics was pretty much stuck. We had all kinds of huge problems. There were very uncertain diagnostic boundaries. We did not know, schizophrenia is not like many other areas of medicine where you have an objective test to say, yes, the disease is present, or no, it's absent, or it's present, and this is how much one has. We don't have anything like that. The diagnostic process is something which we know is relatively reliable. Clinicians tend to agree. But we don't have access yet to any fundamental biology to tell us whether this person does or does not have schizophrenia. In addition, the genetic architecture, the number of genes, what the effect sizes were, how common they were, what we're looking for is completely unknown. We only have absolute guesses as to what that might be. There's also the fact that, presumably, if we have 100 people with schizophrenia, they may not all have the same illness. There may be several different types of schizophrenia embedded in that. And we did not have any idea as to what that was. In addition, a lot of the studies that happened before would look at a few markers in one gene and miss the vast majority of genetic variation in the human genome. The sample sizes were really, really small often, and there were certainly worries about publication bias and all sorts of other problems. Frankly, the field was kind of a mess. And so then we applied, and this was something I learned when I was here. This is, of course, number 8 wire. We sort of applied sort of a typical New Zealandish approach, which is to sort of sit back and say, right, if it's broke, how do we fix it? And we formed something called the Psychiatric Journal Consortium. So this began back in 2007. We assumed that the sample sizes were actually huge, and that no single group could ever, under any circumstances, actually get the kind of numbers that we needed. Therefore, we needed to have a new model of science. We need to figure out how to collaborate in an altruistic way. We needed to come up with a structure that was democratic, innovative, rapid, and quite transparent. And we had to basically get people who hated each other on the same teleconference and not yell at each other. So we actually had to sort of get things together and make it harmonized. And I'll talk more about that. Okay, so that's the big picture. What do we know about schizophrenia circa 2016? So the first thing, I'm going to go through 3 different types of genetic variation. The first has to do with the exome. The exome is our fancy term for the parts of our DNA. So each of us has something like 6 billion bases. Is that the Americanism? I forget. Is it 6? So 6,000 million bases. Okay, 6 followed by 9 zeros. However, that translates into UKism and Kiwiisms in terms of those numbers. And only about 1% of that actually makes a protein. The rest does a lot of other complicated things. Some parts do nothing. Some parts have very, very important functions, but don't necessarily make proteins. So about 1% of the genome makes the 20,000 basic proteins that we all need in order to function, live, develop, and be human, basically. And exome sequencing is the way in which you actually target that 1, 2, 3% of the human genome and understand exactly what's going on there. This really has been the sweet spot for human genetics. So when you think about typical genetics, breast cancer and the gene BRCA1, if you think about Huntington's disease, cystic fibrosis, etc., all the genes you think about when you think about human genetics, this is the technology or variants of this technology or what have been used in order to actually identify those. So typically what this finds are things that are really, really rare. Way under 1%, 1 in 1,000, 1 in 2,500, even less sometimes. But they have a very strong impact because they tend to kill proteins. And this technology has been applied in schizophrenia. There have been 3 small published papers, 2 large. One looked at 617 parent-affected offspring trios. Another one looked at 2,500 cases with schizophrenia, 2,500 controls. One gene has come out as being rigorously significant, the gene SETD1A. And what this gene does is it's a histone reader, and essentially what it does is there's another part of the way in which DNA coils on itself, and those little coils have a second sort of language attached to it, and that language is quite complicated, not completely understood, and this gene is involved in that process. And that's something that's going to be published before too long in Papers and Press. Other things that have popped up are, besides that, no other single genes have come up. These studies should have been well-powered to find them if they really were there. But the things that have come up consistently have to do with neuronal signaling for calcium, things that are at the synapse, the part of when 2 nerves talk to each other, there's a little physical gap that's called the synapse, and messages get transferred across there, and things that are involved on both sides of that have actually ended up coming up very consistently as being important. To the best of our knowledge, though, there are no single gene really strong effect in Mendelian disorders. There's nothing like Huntington's disease that we've been able to find for schizophrenia. In analysis, behind the scenes, there's something like 10,000 cases and another 1,800 trios, so much, much larger than initial samples. And frankly, these things continue the same pattern. Maybe one gene has popped up and a bunch of interesting pathways, but again, nothing to indicate that there's going to be a very quick answer in this part of the genome. Now, usually there's another type of genetic variation called copy number variation. Usually we inherit 2 copies, one for mom, one for dad, for each gene. Sometimes, however, due to some things to people like me that are really quite interesting, the number of copies can change. You can only inherit 1 copy because one of your parents didn't have that particular gene. Or one parent might have had several copies, and you might inherit 3 copies. And this is called copy variation. So instead of having 2, you tend to have fewer or more. These are actually been quite interesting because these are things that are really quite rare. So typically, if we look at one of these might be found in three people with schizophrenia out of 1,000, but many fewer will be found in people without schizophrenia in controls. And yet the risk actually is markedly increased. So these are rare things that can increase the risk of schizophrenia somewhere between 4 and maybe 20-fold. And in fact, I think if you were to look at all of these, there's about 10 of them that are known right now. You would actually find that people with schizophrenia probably have 1, 2, 3% of people would actually have one of these. And this might be something which might actually end up being important. There's a paper that's going to be submitted soon where we've looked at 30,000 cases with schizophrenia. And again, the same pattern pretty much holds. But the one thing that pops up is common variation. So these are things where the genetic variant is pretty common. Most of us have some of these. Many of us have many of these, and we may not even have schizophrenia. So these are things that are common in the population. There are many of them, but they have pretty subtle effect, pretty soft effect. I'll talk about that in a second. This was a paper we published a bit less than 2 years ago in Nature. It had 302 authors. One of my friends was the person that had to type in all 302 names. About 12 hours into the process, he sent me a one-word email which just said death. And then we found out he didn't have to do it, so that was pretty good. But in this large consortium that we talked about, the Psych Genomics Consortium, the PGC, we were able to pull together around 37,000 cases and 113,000 controls. This was truly a global effort. It had something like, there were 52 different samples were actually involved from all over the globe. And in the end, we found 108 places in the genome, 108 independent regions, which actually had genetic variation which predisposes schizophrenia. This increased the prior total by a factor of 5, which was a paper that I did. And so basically, this was a huge step forward for our field. And what are these things actually, what are the genes that became involved? Well, one of the ones that was the most interesting was DRD2. DRD2 is my code, genetic code for the dopamine receptor type 2. So this is a gene which binds, it's at the synapse, the place where nerves go from, information goes from one nerve cell to the next, and it binds dopamine. As it turns out, this exact protein, DRD2, is the target for all effective antipsychotics. And so this was sort of a really interesting synergy where the protein, the gene that makes the protein, where all effective antipsychotics work, was also one that popped up in our screen. Things that are involved in NMDA receptors, multiple things popped up. These different genes, the proteins actually touch, they do things together. The same for calcium signaling. These two genes, the names don't matter, but they actually are part of a really important way in which nerve cells function. It has to do with calcium going in and out of the cell. This particular gene makes a protein which is the hole that calcium goes through, and this guy is actually touches it. They physically interact in relation to their function. And so again, that we're getting these things that we know fall into the same pathway or the same idea, I think is a pretty important validator. Right now, there are 60,000 cases in total, so the study we published has 37,000. There's 60,000 right now that one of my colleagues is writing up, and this has increased the number of associations by 50 more. So now there's about 160 associations like this. And in fact, as it turns out that where we are right now, every time we get 1,000 more cases, we get 4 more associations. And that relationship has held for about 3 years now. Okay, so what do we know about schizophrenia? First, the genetic architecture is my shorthand for, okay, how did nature build this? How did nature build risk of schizophrenia? How many genes? How common are they? How big is the effect? The number of genes are probably in the high hundreds. How common? Most are actually quite common. A few are uncommon or rare and confer large degrees of risk. The effect sizes are mostly weak, odds ratios of 1.1, and a few are actually quite stronger, odds ratios of 4 to 20. Now, when I say 1.1, let me explain what that means. That's a relative risk. So essentially what this means is having that variant increases your risk of schizophrenia by 10%. If the rate of schizophrenia is 1%, 10% of that is 0.1. So having that variant would increase your risk from 1% to 1.1%. So the absolute risk is really, really small. But the relative risk is highly significant. Okay, so this is a critical thing. This is a pretty weak effect. Whereas there are other ones, so some of the stronger ones, you would find in 0.3%, 3 in 1,000 cases, but essentially no controls, or very, very few controls. And that, of course, means that it's rare, but the risk is higher. How do they act? They tend to act additively rather than any other way. This is, again, sort of in the genetic fine print. One of the things that people have wondered about is, do we find genes interacting with genes, genes interacting with environment? These relationships are suspected, but they certainly are not proven, despite a fair bit of work. Okay, what's common? And this is getting at the agenda. So when we talk about common variation, again, the whole notion here is that a precise understanding of the genetic factors, the environmental factors, how they interact, and what they tell us about the biology are really critical to preventing schizophrenia in the first place, primary prevention, and tertiary prevention, coming up with better therapies. And the Psychiatric Journal Consortium, this big thing that we started in 2007, again, we thought it was a really safe bet, that the sample sizes were going to be huge, too large for any single group, we needed to cooperate. If you go to the website here, pgc.unc.edu, you can actually find out a lot about the PGC. We've set it up so that the results of our studies can be downloaded by a single click. And so it's again, we encourage people to use this. We know that these data have been in at least 125 publications, and that's the kind of thing that we encourage. Any sort of responsible or sensible use of this, we want people to do so that we can learn as much as possible. We've had 2 grants so far, and we just got a really nice score in the third one, and it should be funded at any time. And this is going to really fund and push the agenda that I mentioned. So the PGC currently has, let me just describe it to you real fast. So I chair the coordinating committee. I am your prototypical cat herder, whereas my Dutch colleagues like to say my job is to keep the frogs in the wheelbarrow, which essentially means I just write a ton of emails. It's really quite boring sometimes. But anyways, it's what has to be done. The PGC currently has 930 scientists. So I think right now we have something like, people from 45 different countries and well over 200 institutions. So it's truly a global network. We have 9 working groups: attention deficit hyperactivity disorder, anorexia nervosa, autism, substance use disorders, bipolar disorder, major depression, OCD, Tourette's, post-traumatic stress, and schizophrenia. In addition, there's a cross-disorder group where we try to understand, okay, what are the things that make these disorders similar versus different. There's a copy number variant group, and then of course there's a very key statistical analysis group, which is sort of the backbone of the whole thing. We've got a provisional Alzheimer's group as well. But one of the key things, of course, is that psychiatric disorders don't breed true. Comorbidity is ubiquitous. If a person has one psychiatric disorder, they often have another. Very informed and well-intentioned clinicians can have very long debates as to whether that patient has schizophrenia or whether they have bipolar disorder with psychotic features. This is something which is a known issue in psychiatry, where comorbidity is certainly sort of a key feature. And it has a lot of implications for how we diagnose people, how we set things up. And one of the things we're finding is that the genetic basis of these disorders is not pure. They tend to sort of overlap in some very complicated ways. And that mirrors what we've been seeing in family studies for a long time, where with people with schizophrenia, they tend to have more relatives with bipolar with schizophrenia, but also with bipolar disorder and depression. And so it really kind of runs together in some complicated ways. And this is where we are right now in terms of the sample sizes in the PGC. So these are the number of thousands of cases, 10,000, 20,000, etc. I've listed the 9 disorders, ADHD, anorexia, etc. Here's schizophrenia, substance use disorders. And what you're seeing is the smaller bar, the lightest color, was in the first wave of our study, 2009 to 2011. This was sort of an immediate second wave, to 2011 to 2013. This is the current sample size, and this is our projected sample size, what we think we're going to get in about 3-4 years. So we initially started with about 30,000 total cases, expanded to 114,000, and we should have about 500,000. My goal for all the groups in the PGC is for each group to have 100,000 cases. And it looks very strong that these are the actual sample sizes right now. Major depression is very likely to get there. Schizophrenia is going to get there. Post-traumatic stress disorder just landed billionaire, and so I think they're going to make it too. And the orange bars here are showing the groups that actually have multiple genome-wide significant, multiple hits, multiple associations for a disorder. I'm going to go off record here. Major depression is 9, and that's something that we're writing up right now. And I mentioned that my colleague is writing a paper right now, 160 for schizophrenia, bipolar is 20 plus, et cetera, et cetera. So it's really starting to move because we're finally getting the kind of sample sizes that we need. And this sort of shows the relationship between the number of cases and the number of findings we get. And so this is inflammatory bowel disease. And they had every advantage you can think of. In a modern industrialized society, there aren't a lot of things that will give you bloody diarrhea, except for inflammatory bowel disease. And so there aren't a lot of copies or phenocopies that we have. They can make a relatively precise diagnosis because in most countries people get a biopsy and you can certainly tell what a person has. And their architecture was very favorable. They got to about 100 findings with only around 5,000 cases. So that was because the architecture of IBD was very favorable to this. Now, in most other situations, you basically, for bipolar disorder, you see that nothing happens for a while, then it turns a corner, and then starts to increase. For schizophrenia, and this is including unpublished data that we've partial analyses along the way, we didn't find anything until about 6,000 cases, and then it really started to decline and become linear. And this is what the people that study height have found too. And so it's a relatively typical thing, that these common disorders are characterized by a lot of, the main thing that they're about are a lot of common variants of very subtle effect, and you need very big sample sizes to find them. So where does it stop? And so we've got the number of cases and the number of associations. And you have an initial area where nothing happens. And then people write review papers and tell you that what you're doing is stupid, blah, blah, blah. Occasionally, if your architecture is very favorable, they'll take off with very small numbers of cases, but that's atypical. There's a point of inflection where things start to take off and you start to get 1 and then 6 and then 22, etc. But then it actually becomes linear. So like with schizophrenia, every time we add 1,000 cases, we get 4 more. That's the slope. Eventually, it'll tail off when we get close to the true number of associations. It won't go up forever because there aren't an infinite number of places in the genome involved in schizophrenia. And obviously, you get to a point of diminishing returns where you increase things, but you find out a lot of effort to increase, but you don't find out that much. But we don't need to get here. We just need to get there. We need to get to a point where it's good enough, where we've tagged most genes at least once, where we've actually identified most of the pathways, 80% of the time. That's what we need to get, and that is an accomplishable end. And frankly, this is actually kind of remarkable measurement. So I think we actually know what we're doing. We can actually define, what is the kind of study that we need to do. We can actually come up with estimates as to what the sample size is. And that's really amazing for us. Because after having wandering around and not really knowing what we're doing and trying a lot of different things and getting frustrated, it's amazing that we actually have a path. Okay, so I think in terms of the agenda, the first goal is to complete the parts list. We need to find as much about the architecture as we can. how many genes there are, how common they are, what are the effect sizes. This will be the thing that will tell us the biology. There's plenty of examples of a genetic variation that points to a gene, and the variance is very subtle. It's not a big effect at all. But that gene is actually really critical for drug targeting. So for example, the HDL receptor actually is one example of this, where it actually has, for lipids, it actually has a genome-wide significant association that's very common. It's not very large. It's not very, the effect size is not huge, but yet that obviously is a very critical target for drug development. And so the drug companies are starting to pay a lot of attention. And so this is actually a key step to both primary and tertiary prevention. So why do psychiatric disorders overlap? And this is a shot of my son Brendan with his St. Andrew's uniform on back when he was a new entrant. And he had a calculator there, and he actually went on to study mathematics. And he actually went on to develop a key method that we're using right now. He did all the math and all the stats and the software programming to actually make this cool method called LD score regression. And what it allows you to do, it allows you to take the genetic results and look at correlations between two different disorders. And so instead of looking at like a phenotypic correlation, like just looking at, if you study 1,000 cases and 1,000 controls, how often do they have both disorders? You can actually look at DNA and look at the results about DNA and ask the questions from a genetic perspective, how similar versus different are these two disorders? And what I'm showing here is what, this is from a paper that's going to be submitted soon, where they looked at, where we looked at about 40 different neurological and psychiatric disorders. And this is what you get in neurology. So, most of the things to look for are blue-colored squares with an asterisk in them, because those are the ones that are significant. You find that migraine with aura, and migraine without aura tend to be highly correlated. Different subtypes of stroke are highly correlated. Different types of epilepsy are highly correlated. But if you look at all the other ones, there are not a lot of intercorrelations. So you don't find migraine overlapping with multiple sclerosis or with Alzheimer's. And the reason this is important is this is what happens when you actually can diagnose things relatively cleanly. Neurology is in a much better perspective than we are in terms of coming up with diagnoses for those things. But if you look at the psychiatric disorders, they're really, this is a lot of correlations. And so, for example, if we take these one at a time, schizophrenia is correlated with anorexia nervosa, with OCD, with bipolar disorder, with ADHD, and depression, and it's not associated with these two guys because their sample sizes are too small. If we look at bipolar disorder, similarly, it's highly correlated with a lot of different things. And this is one of the key problems that we have in psychiatry, is that the genetic basis, we know that comorbidity is everywhere, we know that diagnostic precision could be improved, and now what we're finding is that the genetic correlations are really, really high. And there's a couple of things within here which are actually really quite intriguing, which are, first, a positive genetic correlation of anorexia nervosa with obsessive compulsive disorder, which kind of makes sense. I mean, people with AN clearly do have a lot of obsessionality about a lot of things. But the two that I point out to you is the correlation between schizophrenia and anorexia, and schizophrenia and obsessive compulsive disorder. This is important because people with these two disorders do tend to have thoughts, beliefs, and perceptions which really are not in tune with the reality that other people experience. And typically, we've labeled those as being overvalued ideas, but we rarely have thought that they actually were truly akin to schizophrenia. And I would argue that these data suggest we need to go back and revisit assumptions like that. So this is something which I think is really it is something we're looking at really carefully, and I think it's something that will help us understand more about the fundamental ways in which we classify psychiatric disorders. Okay, rare variation. So rare variants are things that are quite rare in the population, but they have strong effect. Now, I've said already that schizophrenia is mostly a common variant disorder, but what I really should have said was that it's mostly a common variant disorder. That there may be a way to find single genes of strong effect. This is important because this is a sweet spot for biology. There's a ton, if you actually have a clear connection between schizophrenia and one gene, there's a ton of different things you can do almost immediately in any neuroscience lab. And so what a lot of groups are doing are just going really large. And in fact, if you do the power calculations carefully, you find that the sample sizes you need are enormous. 50,000, maybe 100,000 cases to actually try to begin to identify stuff. But the question, of course, is can we do things, number 8 wire-wise, that are sort of smaller and smarter? Here's one family that we came across with Joe Piven, who's an autism researcher at UNC. And he came across this sperm donor. And the sperm donor's sperm has been used something like 80 times in the States. And the risk of autism to his children is something on the order of about 30%. We were able to find 7 pairs of moms and affected male offspring. And we've actually put them through a lot of stuff. I actually think I know what's causing it. I need to wait for one verification step. But it looks like this might actually be a single gene that he passed on to these 8 male offspring that might actually give them a strong risk of schizophrenia. So again, to do this experiment is only about $20,000, not the tens of millions that doing sequencing of 100,000 would actually cost. The other thing we're doing that I think is kind of cool is we found a way to find the sickest of the sick. So there's still a state hospital system in the States where they've got up to 1,000 people who have highly treatment-resistant psychosis. The first person in the study was hospitalized before I was born. And we've got 100 samples already, and we're actually getting whole genome sequencing on 3 really atypical people. And here's one example: a 40-year-old man who was diagnosed with schizophrenia at age 21, and now at age 40, he basically is incontinent, incoherent, mute, and he's just kind of gone. And obviously for schizophrenia, that's an incredibly marked degree of deterioration. And so obviously we're throwing everything we can at this guy to try to understand what might have happened. Next, in terms of the agenda, is we've got a project called SUN. And the basic idea there is, as I've mentioned, if in fact there are hundreds of genes involved in schizophrenia, how do we understand this? I mean, what is going on there? And our best understanding is that it's probably a network, that there's one or several biological pathways, each comprised of dozens of different genes. And what's happening is misregulation. So it's not the changes in the protein itself, it's changes in the amount, in how they respond during development, whether the right amount is made at the right time, yes or no. And this is something that we're actually pursuing quite actively. But this requires a lot of different expertise. And so I'm working closely, I sort of pulled together sort of a network of about a dozen different labs. This is funded by the Swedish Research Council. And I won't go into detail, but essentially we've got a way to look at proteomics in individual neurons. So we can actually look at proteins and to target one protein, and then to look at all the proteins it interacts with, and then that way to try to expand out to find a network. One of my colleagues has this technology called cellomics, where in 96 or 48 well format, we can actually disrupt in neurons. We can disrupt either 1 gene, 2 genes, 3 genes, whole sets of genes. We can actually knock down pathways and then actually measure about 40 different parameters on what's happening in the neurons. If we actually have something that's really well defined, so this is a high throughput tech, this is a much lower throughput technology, but essentially what my colleague can do is this is actually one nerve cell, and the axons are labeled in our orangish color. It's actually red, but it's an orangish color here. All the synapses are here in green. He can actually go in and do single cell recording. He can do just a ton of synaptic biology, and we can change things. So we can actually knock down genes, overexpress genes, and see what happens within this very well-defined construct. And so this is the kind of thing that we need to do as well. So in addition to come up with to find the genetic findings, I think we really need to sort of collaborate widely. But to do the downstream biology, requires a lot of other, it requires many other technologies as well. This is something that we just started about a month and a half ago. This is a European Union project called Coulson. And I was able to pull together 8 academic labs plus 2 companies, Roche and Lundbeck in Europe. And the whole goal of this project is to find an effective treatment for one person. And so we're beginning with, we'll pick individuals who have either two of these three disorders: intellectual disability, autism, schizophrenia. They have to have what is clearly sort of a causal synaptic mutation in a synaptic protein, and a protein that's at the place where two different nerves come together. Now, there's a bit of magic here, and there's more about this in the next slide. We can take a sample right now of any divisible cell in your body. Blood, skin cell, you pluck a hair, you get the hairs at the root, you pee into a cup, there's still divisible cells there. If we can get a cell that can divide, we can make that into a stem cell. And then we can make that stem cell into neurons. And so this is the whole iPSC, or induced pluripotent stem cell technology. And that's what we're doing. So we're going to find an individual. We will make neurons from that person. We'll distribute them to 8 different labs, academic labs that are working here. They're all synaptic labs. They will all work together to figure out what's going on. And then the drug companies will, if we're successful in finding pathology, the drug companies will be able to take that model and screen it against 10,000 compounds. And the ultimate goal of this is to eventually repurpose an existing drug. So ideally what we would find is we'd say, right, we can show in a dish that for you, this drug has potential benefit, because the synaptic defect that you have is reversed by it. Then we would propose basically an N-of-1 trial for that person. So again, this is precision medicine, but the ultimate goal is, can we even help one person with better therapy? And there's we're going to do 40 people over the course of this project. A friend of mine, Debbie Levy at Harvard, has a great example of this. She's identified 2 people, distant relatives, who have 3 copies of a gene called glycine decarboxylase. Big name, big mouthful. But essentially, what glycine decarboxylase does is it takes the amino acid glycine and gets rid of it. And so these people actually go through glycine more quickly than they should. One person has bipolar disorder, the other person has schizophrenia. And Debbie has shown that under double-blind conditions, if you give these 2 people glycine, their symptoms get better. And under double-blind conditions. So I don't know where that paper is, but it should be coming. But this is an example of what we're trying to do. Giving glycine won't help anybody else on the planet, probably. But for these 2 people, it probably makes a big difference. And that's the sort of thing we want to try here. And this is just to show some of the magic. We've got a German, Oliver Brustley in Bonn, who has a thing called the stem cell factory. And you definitely want a German doing your stem cells. But he essentially has, you put in a divisible cell and money at the start, and you get out neurons at the end. And most of the steps in between are automated. So it's pretty cool technology. And this is just some geeky stuff to show that it actually works. Okay, what does this mean in the end? And let's go back to the goals. Again, primary prevention, tertiary preventions, stop schizophrenia developing, better treatments. And really, the key thing is a precise understanding of the biology. The genes we identify will tell us the biology. That's the whole point. And there's something I haven't mentioned, and this is an extremely important topic. And there's certainly a ton of local expertise here with Martin Kennedy and his colleagues on pharmacogenetics. This is really more about tailoring medicines. So for example, if you have a particular disorder, can we give you a medicine which is more effective at the outset, as opposed to messing around 2 or 3 times? This is a big deal in cancer, for instance, where you really want to make sure that you tailor a medicine to the cancer that a person has. But another big part too is adverse drug reactions. So as you know, many times the medicines we give actually can be toxic, and one of the goals of this is to minimize harm. Can we identify people that are likely to react really, really badly to a medicine? They might start sloughing their skin or something. And again, this is a key part of tertiary prevention. This is not the work that I really do, so I haven't commented on it, but it really is an absolutely critical part of the whole business. Okay, so in terms of what does this mean? I think it's basically proven now that schizophrenia has an important genetic basis. We've done this for twin studies, family studies, and now we can look at the bloody DNA, and I think it really can't be questioned anymore. But again, and I believe we're making progress, but the stuff we're finding is really quite subtle and not deterministic. I also think it's proven that environment's important. People exposure to stress for a long time, brain injury, infections, drug use, etc., certainly are part of it. In addition, the choices people make matter. Sometimes we make choices that actually don't push us in the right direction, and that, of course, is important too. One of the things which is under investigation right now by a lot of us is trying to understand what the mix of people is in a particular sample. Who are the people? I think it's likely that we're going to find that if you look at 100 people with schizophrenia, a big hunk of them will have a sort of very genetic form of schizophrenia. Some people might have a very environmental form of schizophrenia, and some people might have some combination of this. And this is a major question right now. Can we identify how common those mixes are? But I think it's really important that for most people with this serious psychiatric disorder, the stuff that I'm doing right now is not now clinically relevant. Okay? We're making progress, we're learning a lot of stuff, but it's going to take time for this to actually feed back to the clinical sphere. But there are a couple of exceptions. I think that genomics should be part of the initial workup for intellectual disability and autism. That's pretty clear right now. I'm certainly looking at copy number variation and exome sequencing. You clearly has substantial yield in these two disorders. I think for schizophrenia, if you look at copy number variation in a characteristic way, you'll find that something like 3% of people probably have a CMD. And this is important, I think, because some of these have physical comorbidities, and we may need to treat them in different ways. And the other one, of course, is if someone has a dominant form of Alzheimer's disease. Those are probably the major ones where I think genomics really are indicated as part of clinical workup. But this is under investigation, and if I come back in 3 years, I might end up saying something quite different from this. But really, the main implication, I would argue, is hope. Honestly, for the first time, I really do think we know what we're doing. This is now a major initiative in psychiatry. There's a lot of groups working on this. We've been able to attract a whole bunch of real first-rate neuroscientists. At last count, there are 3 Nobel laureates actually working on psychiatric disorders. That's amazing, because that really has never happened before. A lot of the drug companies are actually part of these efforts. They're getting back into the CNS business. Honestly, 10 years ago, most companies were completely out of the CNS business. And working with drug companies is always kind of a mixed bag. But the bottom line is we need medicines. And if we identify targets, and those are things that they may be able to capitalize on, and that might circle back as well. So I think a lot of us anticipate that there's going to be real progress in the next 10 years. And then I close with my thanks. I've been really fortunate to have great teachers, starting with certainly Professor Joyce and Martin Kennedy. I've got a couple of great labs, both at UNC and at Karolinska, a lot of wonderful colleagues. I've named the 900 people in PGC, but that would go until about 10. Swedish Research Council, in particular, I want to thank, but there's been a lot of people who have been funding this work. So my thanks to you as well for your attention.
Thank you very much, Patrick. We've now got 10 minutes or 12 minutes for questions, comments for Patrick. Becky, you'll know about genetics.
Patrick, you stressed that schizophrenia is a genetic and environmental, has a genetic and environmental basis. With your collection of 500,000 cases, are you collecting any environmental data characteristics with those cases?
That's a fantastic question. And there are, the problem though is that many of the environmental risk factors are difficult to assess. So if I'm asking somebody who's 50 years old, who's got schizophrenia, they've been seriously mentally ill for a very long time, if I start asking about what happened to mom, second trimester in utero, that's not a higher yield thing. There are some ways you can get around that. So first, it's possible to do antibody screening, because the infection side is certainly part of it. We're launching, with Bob Wilkin at Johns Hopkins, we're launching a major push to do a lot of that very soon. But there's several countries in the world where they actually do collect automatically a lot of environmental data about what happened to people. And those countries are Scandinavian countries. And that's one of the reasons why I spend half my time in Stockholm. So yeah, so we're hopeful. So we actually have, you know, birth events, we have APGAR scores, that kind of thing. And it's going to take a while to get the sample size we need to do this, but we're working on that because I think that's going to be crucial.
Okay.
It's wonderful.
How are you
That
doing?
was a great talk, and I agree with you about the hope. It's really exciting what we're doing. Terrific. My question relates to the sort of evolutionary processes that have brought about this possibly 500 genes that exist there. I don't know if that's the kind of number you're thinking. But presumably these 500 genes have been brought about in human DNA because of the power of evolution.
Yeah.
So these 500 genes are giving us something. So it sort of leads to the question, schizophrenia is the price we pay for ... What's emerging in your mind?
Yeah, that's a wonderful question, and there's a lot of people that are keenly interested in that particular topic. There was a paper that you may have heard about where a group at Vanderbilt talked about depression being something related to Neanderthals. I'm super skeptical of that, and we're going to try and repeat their analysis, but I doubt it's true. Anyways, so here's the thing. In order for evolution to act on something, typically, you know, for a single variant, it has to have a strong effect. Right? So in other words, if you inherit something that is strongly deleterious to your survival and or reproduction, it's not going to go very far, right? It'll probably die with you, almost literally. The problem, though, with the things that we're talking about is the effects are ton subtler, way subtler. There's no way evolution can act on something which increases risk by 10%. That's just far too slow. People have wondered about whether evolution might act not just on one genetic variant, but actually on many of them simultaneously. In other words, if you inherit, 30 different variants that give you a markedly better chance of doing something, or you inherit some characteristic because of that set of variants, then maybe that's the thing evolution acts on. But the tricky thing is, to actually analyze that requires massive sample sizes. It's not straightforward, not in the slightest. So I'm not sure. But some of the things we're looking at is we try to look, for example, is what's the correlation between schizophrenia and a variety of personality traits or intelligence or that kind of thing. And some of those things are coming up as being kind of interesting. So for example, there's a positive correlation between bipolar disorder and educational attainment. So in other words, the genes that predispose to bipolar disorder are also predisposed to doing better in school. And from having conversations with Peter about this over the years, given his interest in bipolar disorder, you certainly see a lot of people who are very successful academically, as well as in the corporate world or whatever. We know these people, right? Who don't have to sleep a lot, they have a ton of energy, a ton of active. They're not manic. They never get manic or bipolar disorder, but they got something. So I think the more likely thing is that some of these things, if you inherit a subthreshold variant, might actually be advantageous in certain characteristics. But again, it's just a notion right now, and I can't prove it.
Right. One thing that comes to mind is creativity or left field thinking. Often when people have breakthrough ideas, they're called mad, aren't they? You must be mad, and then 20 years later, the idea's right.
Yep.
Do you think it could be in that area of...
Sure, and there was a paper that actually looked at this. There was a paper that came out of Iceland looking at creativity and schizophrenia and bipolar disorder, and they claimed to have a small positive correlation. But the effect was really trivial. One commentator said that if the distance between me and a creative person is a mile, their effect was 13 feet. Okay, so it's really, you can find these relationships, but honestly, they're really soft.
Thanks for that talk. When you've got all the biological pathways laid out for schizophrenia, do you think you're going to have a better, will they help you to find or work out what the environmental factors are with greater precision?
That's a great question. So the question Martin asked is, if we actually have the biological pathways sorted out, will they help us with environmental effects? And I think there's a fair chance it might. I mean, some of the environmental effects that we're talking about are things which could be modeled, right, in a cell. So for example, you might be able to take a human being who has, inherited a ton of these risk alleles, make iPSCs from them, and then actually, induce the environmental stress in some way. What that is depends on the stress, right? There's some that may be inducible, there's some that might not be, but you could see what happens. Or you could take human cells, put them into a mouse brain, and then hit the mouse with the environmental stress, and to see what happens, for example. There are some things that might be possible. There was an example of this in Nature where Fred Gage and some other colleagues at UCSD did a study, the environmental effect was lithium. And they were able to show that neurons derived from people with bipolar disorder tended to have abnormal firing patterns. And they normalized with lithium only if the person responded clinically to lithium and not if lithium did not help them clinically. So that's an environmental effect. But if in fact that observation holds up, that could really revolutionize a lot of things about how we understand the mechanism of action of lithium and why it works in some people, not others.
So I have a transport question. Drug companies partly like to look for common disorders for which they can get lots of people prescribed their drug so they can make any money. Schizophrenia is already 1%, so it's all at the margins for drug companies. But if it's only 3 in 1,000 of people with schizophrenia who have a glycine problem, you're into really these rare and orphan drugs, and it becomes a real risk that drug companies just won't invest the money to ever find the compounds. How much are you getting resistance from drug companies to finding very uncommon, different metabolic pathways in a sense, because there's no money to be made out of uncommon disorders?
So two parts to that, Peter. First, I think there are obviously, there's the established the drug companies that have been around for 20 years. That's one thing. But there's a ton of startups. And so Angelman syndrome, for example, there are 2 Nature papers on this. There's several groups that are going crazy about this. There's a couple of small, high energy, high risk, high reward groups that are actually companies that are working on this right now. Angelman's quite rare, but if you can actually reverse it, that would be a big thing. And the reason why, from a marketing perspective, yes, it's uncommon, but if you have it, you've got to take the drug every day for the rest of your life. And so, and even some large companies do this. Roche, for instance, has a big market. They might have scrapped it recently, but they certainly a couple of years ago, this was a big thing for Fragile X they were working on. Fragile X is uncommon, but if you find an effective therapeutic for Fragile X, a kid with Fragile X is going to have to take that drug every single day for the rest of their life. Right? And depending on your margins, that actually can be profitable. I think would be their argument.
So they don't want a one-off drug that you take and it fixes things.
No.
They put in something that they can go on and off to a few people.
Exactly. So many of you probably know Nick Martin from Queensland, QIMR. Nick said that he's a human geneticist, and he said the most attention he ever got on a paper they did was when they identified a genetic variant which had to do with the curliness of your hair. And the L'Oreal actually had he and his colleagues over in Paris. Actually, it's also the same gene which I believe controls curliness of hair in sheep, doesn't it? It's a really critical, it's an interesting cross-species thing. Graham Montgomery is a Kiwi who actually works with Nick. He knows a lot about this. Anyways, but L'Oreal was all over this because they were like, cool, a woman would have to take a pill every day to make her hair straight, right? So this was like, this is awesome. And of course, it would probably, curl or uncurl your cilia or something, messing you up in some other critical way.
Time for maybe one last question or comment, or else we could say thank you very much for excellent talk. Great hearing that I think it's the hope from the last 5 years of research, and thank you very much.
Just thank you.